for our more creative names for our papers. All right, so I'm going to be talking today about a idealized kind of minimal model, more toy conception of what we would think about as respiratory odor navigation. The history of chemical navigation in A-life is, one that I don't need to preach to the masses. There's a long thing of chemotaxis and simulating moths navigating odor plumes, and it's a very rich area. Chemical navigation itself is, a very, very important and critical adaptation that organisms need to be able to solve, whether it's to, forage for food or find out where their mates are. They have to be able to do it robustly, and they have to be able to react real time in different environments. And not only that, but it's also very different depending on what your body is like, right? So we can see this handsome fellow here with his nice little plumose antenna, and depending on, the different environment, we could also see a cell here climbing a gradient, a very turbulent odor plume, or a very slowly diffusing odor plume, and the way that that environment also reacts with your body is going to be different in different cases, right? So something really strange happened when people started, or when the things kind of crawled out from the water, got backbones, and closed off their circulatory systems, and they started doing this thing, sniffing. What is sniffing, right? We all do that a lot, but it's very strange when you think about it, especially because, it requires that the organism has to actively modulate its own physiology in response to its environment, right? So not only is the environment moving and, blowing around, the chemical landscape itself can be discontinuous or very smooth, depending on the structure of the environment, but you also have to modulate your own physiology in order to figure out what the best way of navigating that environment is. So one of the questions that we're going to be talking about, or what I'm very much just going to be giving a quick kind of sales pitch for you today, is how does this work, right? What are the kind of, if we take a very toy and minimal approach, what are the kind of basic setup that could help us, what are the kind of basic foundations that we can build and scaffold about our understanding for this type of behavior? So what we're going to do is we're going to focus on a very simple case. So we're just going to focus on that oscillatory nature of the respiratory system. And we're going to put this on a very smooth environment. While, of course, turbulent environments and moving environments are naturally very interesting, we're just going to start from and scaffold our approach very slowly. So we're going to start from a more simple kind of case, okay? And we're also going to be having very simple agents as well. So again, it's going to be it's going to be a single chemical source. We're going to evolve circuits of continuous recurrent neural networks. For control, as a proper control population, we're also going to have agents that are just regular run-of-the-mill chemotaxers, right? They're just sense the exact position or the exact concentration at their position. But we're only going to be comparing them today in the evolutionary plots, but which I'll show you in a second. But for our respiratory agents, they, of course, so each one of them, both the continuous and the respiratory, we say have two nares, if you want to think about them like that, right? They have two nostrils. And they also have a designated, for the respiratory agents, they have a designated neuron that acts as a kind of, quote unquote, phrenic neuron, or something that's in charge of the way that we modulate our physiology, right? So what is it about this that we're trying to capture, right? What is this respiratory sensor and how does it work? So we're just going to walk through, it may not make sense, and I'm going to give a very brief, kind of capitulation of it, but what we wanted to capture was just the way that when you're sniffing, you can only get things on the inhale. You have this inhalation-exhalation process that is continuous and being generated by oscillations in your physiology. So we have some sort of mathematical relationship in order to capture that process. We also, inside of this inhalation or respiratory phase, we have a breathing rate, which is simply bounded to a certain way. So it can only have either, a sniff every 10 seconds or a sniff every, 2 sniffs per second. And the way that it does that, the way that it modulates the speed of that is through the output of that phrenic neuron that I just mentioned, that third neuron. And we do this for very simple circuits. So again, we evolve 3, 4, and 5 neuron circuits to do this. And also, if any of that, I'm blowing past the math very quickly, but all you need to know from it is that the agent can only detect the chemical when it's inhaling. It can modulate how fast that it's sniffing or breathing through the output of that phrenic neuron. And if it's breathing very quickly, importantly, it can't do so forever. So I'm not going to be able to talk about the kind of constraints that we, in the interest of time, the kind of constraints that we put on the system. But basically, like real organisms, we have to balance the amount of oxygen and CO2 that's in our bloodstream in order to, that's why we can't, just sniff constantly, right? Otherwise we'll pass out. So this is what we discovered. So I just want to point out very quickly that obviously respiration is much harder, right? It's just a more difficult version of the chemotaxis task. So this is kind of at least to be expected. But they still nonetheless were able to, I keep on forgetting this is a touch screen. There's still nonetheless a good portion of agents that are able to solve the task robustly, right? So what is one of these? So the plots on the right too, as well as the agents with the continuous sensor. The plots on the left are the ones with the respiratory sensor. So we can see, but also I would like to point out that in these fitness over the generations plots, these line plots, the x-axis is different for the respiratory sensor. So it takes 1,000 generations in order to get there for the respiratory sensor, whereas it only takes 500 generations, and we can see that that's a much more quick ascent. So what does one of these agents look like? So first I'm going to talk about this behavior that's very common in the chemotaxis literature, is this kind of casting, right? So both simple cells and animals such as moths or other things do this behavior where they basically, you can almost imagine it as they're like doing donuts in their car when it's snowy outside. They're just kind of spinning around and they're taking in and they're integrating over time to be able to detect that difference, right? But importantly, so also I want to say in the continuous sensor case, this is extremely common. Almost all of the agents basically end up doing a strategy that's kind of similar to this. Although the you can of course, you always have to make sure that the parameters of the system, such as its maximum turning angle or how its maximum amount of thrust, those can also affect the amount of strategies that result. So for an example, you can get more clinotaxis type strategies. So the animals that move their head back and forth in order to detect gradient difference, if you end up playing with that maximum turning angle or thrust, right? But this, at least in our setup, this casting behavior was very common for agents with a continuous sensor. This agent is a respiratory agent, just pointing that out. So also with respiratory agents, we end up finding all right. No, it spoiled it. All right, you didn't see that. So, all right, we also find this interesting behavior that is typically talked about in the literature as being like a kind of this active perception type sequential sampling. There you go. So like many vertebrates, these agents will, oh, that's very unfortunate. Is it going to do it? Yeah. All right. So like many vertebrates, what they end up doing is they'll move for a little bit and their breathing rate will drop, and they'll start to increase their breathing rate when they're attempting to triangulate where the chemical source position is. So what we can see this agent doing is that it's moving, it's sniffing, and then it's moving a little bit more. So it seems that this video is not going to work, so I'm just going to move on. So when we actually end up looking at that breathing rate, what you see is that this plot here on the leftmost side is that it does that sequential active sampling type strategy. And this is something that only emerged in the respiratory agents. Pretty interesting, right? So another thing is that this behavior, while it's very present, is also a part of a much larger diversity of behaviors. So we can still see that while there's still casting methods present, there are still various different types of forms and ways that this both sequential sampling and this casting behavior are realized by these agents. So while I'm unfortunately not gonna be able to talk too much about it today, what we were able to do is we were able to do a dynamical analysis on these agents and try and figure out what was the underlying neural mechanisms that gave rise to this behavior. I'm only going to talk very briefly about the casting strategy. So basically, when we look at correlations between the breathing rate and the motor output, we find that the agents actually have a very positive correlation with their breathing rate and their thrust, and a very negative correlation with their torque, which when we perform dynamical analysis, we find that this is actually true. So what happens is that agents that use that spinning, they're doing donuts, they end up, in order to find the chemical source, they have this underlying neural strategy of, I'm going to increase my thrust and decrease my torque as my two nares detect more chemical in their environment. And importantly, what we see on this bottom, this triple I here, is that they have to balance their physiology in order to do that. They have to basically, what this agent does is it finds a very low period oscillation, and it continues that low period oscillation while it continues to navigate on the inhalation cycle of its respiratory rhythm. So again, just to go over our main takeaways. So agents were successfully evolved to do that respiratory navigation task. And it is very interesting because, it wasn't really known in the literature that they were able to do that with only the use of a respiratory sensor. So many people thought that that kind of sequential sampling was a result of the other gating mechanisms and result to wind direction, or whether or not they detected a visual stimulus. But we were able to show that simply through chemical navigation alone, they were able to do this, engage in this type of behavior. Also, we looked a tiny bit, not really at all, at that dynamical mechanisms that gave rise to the casting strategy. But it's going to be the job of future work to look into the sequential sampling strategy for these agents. So I just want to thank all of my friends and collaborators, and thank you very much for listening.